Remember, the content on our show is for information only.
It is not intended to provide medical or other professional advice.
Hello and welcome to Placebo, the podcast where each week we try and teach each other something interesting about healthcare or medicine. I'm Logan Richards, and this is my sister.
And I'm Kaylin Richards.
And this week on the Placebo podcast, we are going to talk about superbugs.
Oh.
Not really, though, but
no?
That's the clickbait topic.
Okay.
So
I hate clickbait.
Well, we won't call it that then.
Okay.
Do you know what superbug means?
Of course. Of course.
What does it mean?
It means something that's not easily killed by antibiotics, right? That's antibiotic resistant.
I can't tell if superbug is a media-created term or a science-created term for the media. It sounds like it probably...
Sounds media-ish to me.
Yeah. But basically, whenever the media says it, they're talking about some type of antibiotic-resistant strain of bacteria.
Like MRSA. Methicillin, I think.
Methicillin-resistant Staphylococcus aureus.
Or VRE. Do you know that one?
Not off the top of my head.
I think it's vancomycin-resistant Enterococcus.
So a lot of these, as you'll see already, actually, the bacterial strain is almost named for the resistance to which antibiotic they're resistant to.
Yes.
There's a lot of other common ones. So a good example is C. diff, which is, if you ever go into a hospital, most of the hand sanitizer gel things will have a sticker on them saying that it doesn't work for C. diff.
Yeah, or if you... So all the rooms in the hospital will have a precautions sign at the door. Mm-hmm. I think that stands for all hospitals in the U.S. And so it'll either just be like a universal precautions, which is for anybody.
Right.
Or there are different types. But if someone has something like C. diff, they have enteric precautions, meaning you have to wash your hands with soap and water is one of the things that entails.
Right.
Because the hand sanitizer doesn't kill C. diff.
Okay. So I haven't planned to talk very in-depth about antibiotic-resistant bacteria or superbugs in themselves, but we need to introduce them in order to get to what today's real story is.
Oh, okay.
So the way I want to do that is, I assume that you know the podcast Radio Lab.
Yes.
Okay. So
Maybe.
So they had an episode last year that was called Staff Retreat, which is a play of words, of course.
Oh, what a pun.
Staff spelled the way of the bacteria, not of the group of coworkers.
So a PH.
Right.
On the end there.
So they had a really interesting story. And, of course, they started with this famous anecdote, and it sounds like people are not really sure if it's real, but of Alexander Fleming, who was a British scientist. And in 1928, do you know what he discovered?
Penicillin?
Penicillin.
Aha.
And according to the story, he didn't really discover it. He just happened to have his window open.
This is what I've heard, yeah.
Right. So people aren't really sure if this is true anymore. But apparently he had his window open. He was on vacation. Penicillin floated in as kind of natural mold in the air.
Yeah.
And happened to land on a petri dish that he had left over vacation.
Seems like a really bad scientist.
Really? That's exactly what they said in Radiolab. They're like, this dude was so messy. He just left staph growing. So anyways, he noticed that some of the staph had died, and he figured out it was penicillin. And so he was like... There was all this media attention.
It was not good controlled experiments.
Well, it wasn't really. It was just it was circumstantial. It was a coincidence. But he was able to capitalize on it. He ended up winning the Nobel Prize in 1945.
For his accident.
Good
thing he went on vacation.
Well, he figured it out, right? He had to be the scientist in the first place.
He did, okay.
But here's a quote that he said while accepting his Nobel Prize. "There is a danger that the ignorant man may easily underdose himself, and by exposing his microbes to non-lethal quantities of the drug, make them resistant."
Yes.
So he, the creator of,
He saw the writing on the wall, I think, already.
And that was actually interesting, too. I'm going to post the link to the Radiolab episode because they did a much better job of talking about this list of the time that it takes for bacteria to become resistant after an antibiotic is introduced. And so one thing that they said is, as Alexander Fleming was getting all this media attention, there was an article published about strains of Staphylococcus that were already penicillin resistant. So it was kind of, and they went through this big long list of like every, couple of years a new antibiotic would come out, and months or years later it would be resistant, or bacteria would be resistant to that antibiotic.
I know currently there's much fewer types of new antibiotics being found and discovered.
So the issue with that, what Radiolab was said is that, and we all know the cost of making and producing a drug is really expensive. So a lot of pharmaceutical companies are having a harder time seeing the benefit for their company in making and producing new antibiotics, because as soon as they make a new antibiotic, it's going to start becoming resistant. Does that make sense? So let's talk about a couple of statistics from the CDC. Center for Disease Control and Prevention, apparently, but that's not in their acronym.
They didn't forget the P, I guess.
So they say at least 2 million people in the United States acquire an infection from bacteria, and that leads to about 23,000 deaths. I've seen in other sources that worldwide it could be up to 700,000 deaths a year because of these so-called superbugs. So I think, and part of the problem is we're not really tracking those very well. So
I would say it seems a little bit even low to me based on just my general experience of how many patients have that on their diagnosis list.
The CDC, I think
not that people are dying from that necessarily, but
it sounds like a lower percentage of the people that have an infection actually is actually fatal. So, which is, that's good. But the CDC, I think one of their main things that they want to focus on in the future is tracking the disease progression of all these different bacterial diseases. So the number may be low. They estimate that in 2008, these numbers, money numbers I'm about to tell you, are $2008. So $20 billion in excess direct healthcare costs, with an additional loss of productivity up to $35 billion.
What's productivity?
Loss of productivity. Oh, that's meaning because people are spending extra days in the hospital. They're not working.
Not able to go back to work.
And I don't know, it probably depends on what they looked at. But it basically means that someone is not, doing something else because they're either sick or they're taking care of someone who's sick.
Okay.
So maybe, even the doctor who has to see this person, a couple of days longer because they're sick is not seeing someone new, whatever, those kind of things. And that person is obviously not working because they're still in the hospital.
And their family member might be taking time off to take care of them.
Right. Yeah. All those types of things are usually put into that type of measurement. They also say that about 50% of all the antibiotics that are prescribed are what they call not needed or not optimally effective as they're prescribed. So that's just pointing out there's an issue in the way that we are using antibiotics, which of course is how these resistant strains are appearing. It's pretty clear now and common knowledge that because of the life cycle of bacteria being so short, and antibiotics, when you take them, kind of create this toxic environment that kills most of the antibiotics, but sometimes it kills most of the bacteria. But sometimes there's a mutation because bacteria have such a short lifespan and produce in such large numbers. A genetic mutation happens, and those ones that don't die because of their genetic mutation go on to live.
Survive and then replicate rapidly.
Right, as bacteria does.
Yeah.
So that's really the problem that we're seeing here. So in Radiolab, they have this awesome story about how the window is open, and they're like, well, why don't we just, leave the window open and hope something new flies in? And then their story ends up being about, have you heard that episode?
Possibly.
No?
Sounds familiar.
Their story ends up being about this pair of historian and microbiologist, and they go back and create this medieval antibiotic out of weird onions and other stuff that they have to boil. And it actually works for a little bit, which is, so it's a super interesting story. You got to go back and listen to it.
Okay. Are they using like historical medical texts and trying to see if something works?
Yeah, pretty much.
Okay.
Yeah. And they had to so they were translating from Old English. It was very interesting.
Okay.
But they said that was kind of their metaphorical new open window. Mm-hmm. But to add on to the story, we're going to add on a third metaphorical open window.
Okay.
Are you ready?
Okay.
So today we're going to be talking about recent...
Wait, that was all intro?
That was all intro, just so you have
okay.
some
We're 13
background
minutes
knowledge.
in. All right, what are we talking about?
Well, the main story of today is a recent discovery by a 25-year-old PhD candidate.
Oh, you did say I was going to feel bad about myself in this one. Oh, okay.
She is older than I am.
Not me, though. I'm barely older than you.
She is currently studying at the University of Melbourne.
Oh, we talked about Melbourne in our last last episode.
That's the little connection I was telling you there would be.
Perhaps she rides her bike.
Perhaps, hopefully with a helmet on.
Yeah.
Although it seems most in Melbourne do.
Yeah.
Okay, so her name is Shu Lam, which I assume I'm definitely pronouncing wrong.
Possibly.
And she, from what I can tell, is studying bioengineering.
Okay.
And her kind of crazy, although her discovery, as it were, is a new class of basically bacterial infection treating agent.
Not necessarily antibiotic.
That is not an antibiotic.
Not at all antibiotic.
Not at all antibiotic.
Okay.
And
does it have a name?
It does. So she has a nice, I was going to say, a snappy little acronym.
Okay.
And the
Snappy.
reason it's snappy is because it's SNAP.
Oh, brother.
SNAP, there's actually two P's, stands
Snap.
for Structurally Nano-Engineered Antimicrobial Peptide Polymers.
Is it nanobots?
I don't think so.
No.
Okay, so I have a nice little picture to show you. Do you want to describe what a snap looks like?
Is that whole thing the snap? Or
so this is a cartoon representation from their paper of what a snap looks like.
I don't have my glasses, so let's go a little closer here. They look like a bunch of balls of slightly different colors. That might not be important. And there'll be chains of these balls, like maybe 10 or 15 long, all in a line, and then they seem to be connected on one end of their chain to a bigger ball. What do you think? Is that good?
That's good.
Okay. So, it looks like a spider with a lot of legs.
Okay, so in the literature, these are described as stars.
Or a star with a sort of.
So I like to, it kind of looks like a jellyfish to me, or like
a jellyfish, something with a center round shape and a bunch of lines coming off of it.
Okay, so what you're seeing here, there's a central area, and sticking off of it are a bunch of these antimicrobial peptide polymers. So
those are the little balls.
These are all the lines. These little like tentacles almost.
Oh, the polymer is composed of all these little round balls.
So they made a bunch of different versions. The one that's most successful is a star with 16 of these little arms. You don't really need to know that.
I didn't count it, but sure.
This might be a 32 one. It looks pretty big. So what they did, basically there's already a area of research looking at these AMPs, what they call them. So that's antimicrobial peptide, I think. Might be something different, but that's basically it. So one of those things, which is, it looks like a little piece of spaghetti maybe, a little singular thing. So there's already some research looking into those being antimicrobial. Seems like what she's done new here and her research team is put them in this star formation, kind of merge them all together into this squiggly jellyfish star thing.
Spider.
And so what they did is tested them against a bunch of different types of bacteria that are kind of commonly known.
Staph.
Yeah, it sounds like they did.
E. coli.
They did like MRSA, they did E. coli. And what they found is that this kind of combination, these snaps, work best against gram-negative bacteria.
Yes.
Do you have any idea what those are?
Well, I know that there are gram-negative and gram-positive, and it's to do with the type of stain, I think, that they use to identify bacteria.
Right.
and it's like positive if it takes the stain and negative if it doesn't. But I don't know what it indicates on the bacteria itself. Or
Right. Okay, yeah. So that's basically it. Some old scientist named Gram had some stain. He saw that some bacteria got stained by it, some didn't. And so that's exactly what you just explained. The true difference, from what I can tell, is these gram-negative ones have this really unique outer membrane.
So that's why they don't hold on to the stain.
Right, which seems to be super impermeable to a lot of stuff.
I think that whether a bacteria is gram-negative or gram-positive affects the type of bacteria that people, or the type of antibiotic
right, because
will work on it.
You need a different type of antibiotic to get into the outer bacteria, or
if
the
it's
outer
gram-negative, yeah.
Right. So it's actually, from what I can tell, these gram-negative bacteria are even more of a problem when it comes to resistance and than gram-positive. So gram-positive is I think MRSA is gram-positive. So we all know that's a problem. But there's other ones that their names aren't quite as well known in the media, but beyond E. coli, which is gram-negative. But that these ones, because of the kind of the structural makeup of the bacteria, are harder to kill. And so therefore, if you don't take the right dose of antibiotics, are almost at more of an advantage to have some of them survive and then reproduce after the antibiotics are gone.
So you're not going to be better.
Right. So these gram-positive or gram-negative bacteria are harder to kill, and they're actually a bigger problem with antibiotic resistance. So it's really great that these S16, which is what they're calling the SNAPs.
The SNAP 16.
Yeah, it's really good that the SNAPs work against those, because that's actually a bigger problem.
Okay.
So they found that they tested 6 different types. They found the SNAPs work better on the gram-negatives. Then what they did is put a gram-negative bacteria into mice.
I was going to say, I'm assuming this isn't a petri dish to start out with.
Yeah, all of that is what they call...
Or something similar.
Yeah, it's all basically, it's out of living organisms. But then we went into mice.
What did they say? Oh, we talked about this
in vivo
before.
is in mice.
In vitro?
It might be, but I don't know. So then they put gram-negative bacteria into mice, infected them all. What they ended up doing was having a control group that wasn't treated, another group that was given a antibiotic, and a third group that was given the SNAPs, basically. And then what they found is that, unfortunately, the untreated group all died. The antibiotic group all had survivors, but what was interesting, as well as the SNAP group, so both those groups all had 100% survival rate at the times that they were checking.
Okay.
From what I can tell.
So the SNAPs did not harm the mice.
SNAPs didn't harm the mice, seemed to kill the bacteria because no one died. Same with the antibiotics, except when they looked at the antibiotics... and then went in to look at white blood cells and things like that. They were looking for neutrophils in the peritoneal cavity, which is in the intestines.
In the abdomen, yeah.
In the abdomen. So they found that the ones that took the antibiotics had no neutrophils, therefore weren't gaining any type of antibody resistance.
Any immune system
advantages. But the ones that had the SNAPs had a bunch of neutrophils, therefore gaining a little bit of advantage from your own immune system being able to fight the bacteria too.
So in future, if this mouse got exposed to the same bacteria again, it might have more immunity to it?
Right. That's the idea, at least. All of this is very new. So let's go to the next big thing. So basically what you can tell is, and what we know from antibiotics, is they kill good bacteria and bad bacteria, which is kind of that trade-off we always say.
Yes, that's why people have GI problems after they...
Right. You often get told to eat yogurt or something.
Take some probiotics, which we have another episode on. But...
Right. So with these SNAPs, it seems like they're not killing any of your cells, which are interesting.
That is interesting. How is it so specific?
Well, the answer to that, I was either not able to fully comprehend or they've left it vague on purpose.
Okay.
Basically what I can tell is that these SNAPs are larger than is possible to interact with healthy living cells, like the ones in the mice. Mm-hmm. They say that the snaps are about 10 nanometers in diameter, which means they can't enter or interact with healthy cells because they're too big. That's kind of the basic definition that they gave.
But I would think that, it still doesn't make quite sense to me, because I know that, for example, E. coli can be a pathogenic bacteria, something that's causing disease, but it also is normal to have a little bit of E. coli.
It's normal to have staph on your skin too.
Right. So how would the snaps know the difference between the stuff that you want to be there and the stuff that's too much, or
so maybe it does
the wrong balance or something?
Yeah, and that might be a part of the information that I'm missing. Maybe it does kill good and bad bacteria. What they do say here is that basically it's non-toxic to the animal cells.
Oh, okay. So
does bacteria have a toxicity to human
antibiotics?
Cells usually? Yeah, antibiotics.
Yes, especially some of the more powerful stuff that is used on the stuff that is antibiotic resistant.
Okay.
Like vancomycin, I know, is one that's used. It's a more powerful.
And so that has
I'm not a pharmacist, but right. I just know this. It's something that's used with the more powerful antibiotic resistant bacteria, and they have to check levels for it because you can get vancomycin toxicity if the levels are too high in your body.
Okay, so this is that makes a lot more sense to me then. So basically what they're saying is there's very little to no toxicity for the living cells, but it remains very effective on the bacteria. Okay. So probably killing good and bad bacteria, but not resulting in toxicity for your living cells.
Okay, yeah. Some bacteria or some antibiotics like that can have long-term effects on people that have taken it. Okay,
so that's another good thing. Mm-hmm. I think what that means probably in the end is they can give higher doses that might be effective to wipe out a strain of bacteria, and then they won't have to worry about any type of toxicity. Mm-hmm. Okay, so that has to do with the size. For some reason, them being 10 nanometers in diameter is too big.
Too big. They can't get in to human cells or any mice cells or
whatever. Yeah, that's only been done in mice so far.
And we know what studies that have only been done in mice mean for people.
But this is bacteria, right? I mean, it worked we can hope for the best.
Okay.
People are excited about this.
Okay.
So the next thing that is cool about these snaps is that they seem to not lead to any resistance growing over time, over generations.
They can tell.
That they can tell.
So pretty good, though.
In this study, they used the 16-limb star thing of the snaps.
Yes.
And tested it against 600 generations of gram-negative bacteria.
And in typical antibiotic use, we would expect to see some resistance develop in that many generations?
I think so. I don't know, honestly. But I assume that's why they chose 600.
Okay. It's a lot.
That's a good question. Yeah, I don't know how long it takes for 600 generations to go through.
If Alexander Fleming was already aware of it being a problem, it can't be too many. I mean,
right. Yeah. But bacteria generations is probably really quick too.
That's true.
So we don't know. Yeah, we assume that this is good, but I honestly don't know about that since I'm not a microbiologist.
Or how it compares. Yeah.
Right. But it is 600 generations, which we assume is an impactful, important number. No resistance after 600 generations. So the reason they give for that is that there is the way that the snaps interact with the bacteria in order to kill it. They say that it's multimodal and non-specific. What that means, from what I can tell, is that it's doing several things, which are not always the same.
Multimodal.
And it's not always doing them the same way.
So it's hard for the bacteria to respond or adapt to that? Because it's too variable?
So from what I can tell, remember how I told you earlier there was already some research in the singular strands of what they're calling AMPs.
Polymers, right?
Yeah. So there's already some research. It seemed like the problem with those is what they did is they just slipped through the outer membrane of the gram-negative bacteria, went into the inner membrane, and then just created these pores in the inner membrane, which kind of caused everything to leak out. Mm-hmm. And the problem with that is that I guess they're doing the same thing every time. And so just as with antibiotics, doing the same thing every time means that eventually someone's going to be immune to the way that that thing is done.
Some bacterial cell will mutate such that the polymer can't slip through and do that.
And then there's a possibility of developing resistance.
Yeah.
So it sounds like what they've reported is that this is possible with just a single strand, but because of the way that the snap interacts with it, it does kind of different things all the time.
Do they know what it does?
Well, they don't. And that's part of the research
that makes me feel comfortable.
So they know that there's a cascade of things. I really enjoy this animation, though.
Oh, there's an animation.
Well, a cartoon animation. So what we're seeing here is the outer membrane of...
I can't see without my glasses. It's too little.
Well, I have to describe it to you in a way that makes sense anyways for the podcast. So what we're seeing here is a picture of the outer membrane of a gram-negative bacteria. So we see these chains here, which are liposaccharide, like basically
I do remember
arms.
my basic biology. They're like little balloons that are all lined up next to each other.
Right, and this is that biphosphate layer. That's pretty much all cell membranes are made of that.
Yep.
So basically what you see is a wall, and then on the outside of the wall are these, the bacteria has its own kind of wavy liposaccharide tentacle things.
Okay, little molecules coming off there.
So what we see with the amps, which are our single strand polymer, is that they just kind of slip through. Don't cause any disruption in the outer membrane.
Yeah.
Because of the way they interact with it, they can just slip through and go into that inner membrane through this kind of void space down to the inner membrane, which is another layer that's less, it's protected by the outer membrane, so maybe more vulnerable.
Yeah.
These snaps come in, and instead of just slipping through the outer membrane, they come in and kind of just get tangled up with everything because they've got their own limbs, and then this bacteria has limbs flipping around.
So they're just like sailing around, waving their arms around.
As you can see by this picture, they just kind of get in and get caught with stuff and rip stuff apart.
They're just real clumsy. They're
crash through this outer membrane.
They're like a bull in a china shop. Is this the
seems like
really old school metaphor?
And the way that they're moving through is because they're attracted to different electrostatic charges, basically. It sounds like the inside of bacteria are more negative. So they just kind of come through and crash through the membrane.
Okay.
And then after that is a cascade of a bunch of things that they don't really understand. So when all this crashing and stress happens to the outer membrane, it starts to set off a cascade of things that happen in the bacteria that make the bacteria want to self-destruct.
Seems like a bad impulse on the part of the bacteria.
Well, the bacteria knows here that something is happening to the outer membrane.
They've had some major problem with their outer wall.
Right. And so that stress seems to lead to different cascades that lead to the basically self-destruct function of the bacteria.
Yeah, they're in a castle. Their castle's been wall has
all
been
the walls
toppled.
All
so
the
everyone
walls are
takes
broken.
a cyanide pill? Is that what's
I guess so.
They don't want to be taken alive?
Yeah, apparently not. But the reason that they think that there's no resistance from what they think is that because this thing is just slamming through and doing different things anytime, just crashing into the wall.
I was demonstrating.
Yeah, grabbing stuff, breaking things. that there's different cascades at different times, different things happen.
Okay.
All of them seem to lead to the bacterial death, but it doesn't seem to be.
It doesn't try to repair itself or fight it off. It seems weird that it's not.
Well, I think that's just one of the things that they have seen. So they make it clear that they don't know exactly why this is happening, and that's going to be next steps for research. But it seems like the cause of death is not always the same, which is why resistance can't take a hold.
Okay. I still don't understand, though, why the bacteria cell doesn't seem to be trying to do anything.
Well, they don't either. All of that, in summary, basically means they have this thing that is not an antibiotic.
It's a snap.
It's a snap. It seems to be able to kill bacteria that are commonly known to cause these bad infections that antibiotics are having trouble killing. It seems to be able to be given to a living creature without toxicity to their healthy cells. in these preliminary tests.
Although it worries me if we don't know exactly how it's working that there could be problems with that at some point.
Well, this is all too new to know.
Yeah.
And third, or whatever number we're on,
20th.
Yeah, it seems to be somewhat resistant to causing bacterial resistance. To use the same word twice.
Say it again. I'm confused.
It seems to
not cause bacterial resistance.
It seems to not cause bacterial resistance over a seemingly impactful period of bacterial generations. 600.
Okay.
So I guess what the moral of this story is that right now we have a large problem with antibiotic resistance.
Yes, I think we're hanging on by the skin of our teeth with antibiotics right now, but I don't think they're going to keep working for forever. Yeah. And I mean, they're on the verge of not working now for
and
in
large, worldly international organizations like the UN are having conferences. They recently just had one in September 2016 about this, and everyone is very scared, thinking that these, simple things, like one was mentioned as like gonorrhea, which has been, since the invention of antibiotics, this really simple, treatable bacterial infection, might end up actually being lethal if we can't treat it with just normal bacteria, because it's beginning to become fully resistant. There's a quote from the PhD, I guess, advisor, mentor to the lady who discovered this.
Oh, what's her name again?
Shu Lam.
Or
something like that. So he says, a quote from him that was in, I think, the Telegram, so a British newspaper. The really
Telegraph.
To the Telegraph.
I don't think the Telegram is a newspaper.
The Telegraph, so a British newspaper.
Okay.
He says the really good news about all of this at the moment, if you have a superbug and we run out of antibiotics, there's not much you can do. At least you can do something now. So he's thinking, what they've said as a timeline for all of this, they think maybe 5 years they'll be able to do enough research to try this in humans.
To feel comfortable trying it.
From what I can tell. So it's going to take a lot more research, interdisciplinary, looking at, the reaction to this in all sorts of situations. But from all the things that I can tell, like reactions by scientists in the media, is that everyone is thinking this is going to be very important, which, things like that happen all the time. There's a new discovery and people think it's going to be very important, and maybe it doesn't pan out, maybe it does.
Yeah.
But this is important because a lot of the things that we know or hear in the media about superbugs and antibiotic resistance is that we're kind of losing that battle. And this might be the next open window, as Radiolab suggested, to help us get back to where we have been. The reason that modern medicine has advanced so far with these antibiotics is, the advances that modern medicine has made because of the antibiotics is incredible.
Yeah.
And it seems like we're right on the edge at this point to kind of making a step back. So this kind of thing that's not an antibiotic might be the way that'll keep us moving forward.
Yeah. Bacterial infections killed so many people before antibiotics, and they could again if we don't have something else to be a solution.
Right. So let's hope
as
for
antibiotics
snaps.
don't work. And it's got a fun name. Snaps. Your doctor says, I need you to take a 10-day course of snaps.
A snap pack.
Oh, maybe they come in a little pill pack.
Yeah. So
a good suggested marketing name.
All of this is very, I'll call them later.
Snap pack.
All of this is very new. We can hope for more research on it, as I'm sure there will be.
Right.
They mentioned right now
I have to, always hold back my full excitement since it's only in mice.
Yeah, and right now what they're hoping for is more funding. They think they'll be able to go faster if they have more money.
And more people that can therefore work on it.
Usually the case, but right now it sounds like but based on the media coverage and the reaction of the scientific community, I would think that getting more funding is a fairly reasonable task for them at this point.
You would hope.
But we will see. So for now, I think that's it for this episode.
Okay. We need to have a stronger conclusion. Sometimes our episodes just peter down.
Peter out. No, abrupt endings are funny. Just kidding. If you enjoyed the episode, please share us with a friend or give us a rating on iTunes.
You can visit our website, placebopodcast.com, for our show notes, or to send us any suggestions or feedback. If you don't want to go on there, our email address is
placebopodcast@gmail.com.
placebopodcast@gmail.com.
You can also find us on Facebook, or we have Twitter too, but I don't think anyone finds us there.
I'm not sure how Twitter works.
It doesn't.
Thank you to James Aiken for our music.
And thank you to you for listening. We'll be back next week with a new episode. Goodbye.
Goodbye.